Millions of species share this planet, from microscopic single cells dividing in a drop of water to blue whales surfacing in the open ocean. To make sense of that massive diversity, biology starts with a surprisingly simple question. How do we draw a hard line between what is alive and what isn't? If you ever get stuck trying to remember definitions like these while studying late at night, our 24/7 WhatsApp study coach is right in your pocket to help you figure it out. To define life, we use the MRS GREN checklist. If a specimen ticks all 7 boxes, it's living. M is for movement, R is for respiration, S is sensitivity, G is growth, R is reproduction, E is excretion, and N is nutrition. As you look at this diagram of a virus, notice that it fails the test. Because a virus cannot carry out these 7 life processes on its own without taking over a host cell, biologists do not classify it as a living organism. Once a specimen actually passes the checklist and earns the title of living, your next job is to figure out where it fits within the massive web of life. We sort organisms into groups based on the specific physical features they share. For instance, if an animal is warm-blooded, has hair, and gives birth to live young, we place it in the mammal group. But relying only on outward physical traits can lead to mistakes. With advances in technology, scientists now look at the very instructions that build the organism. By analyzing the base sequences in DNA, we get a highly precise scientific look at evolutionary relationships. The rule here is strict. The more similar the sequence of DNA bases between 2 species, the more closely related they are. It proves they share a more recent common ancestor on the evolutionary tree. To ensure a scientist in Tokyo and a scientist in London are talking about the exact same species, biology uses the binomial system, an internationally agreed-upon naming standard. Every scientific name is made of 2 parts. Look at Homo sapiens. The first part indicates the genus, which always starts with a capital letter. The second part is the species, written entirely in lowercase. And when typed out, the whole name must be in italics. Following these exact formatting rules allows anyone, anywhere, to communicate using a precise, universal language. When you sit down for your exam and are asked to identify an unknown organism, your most reliable tool is the dichotomous key. The word dichotomous simply means branching into 2. A key works by giving you pairs of opposing physical descriptions and asking you to pick one. As we look at this flowchart, imagine we're trying to classify an unidentified specimen, in this case, an animal with fur and webbed feet. We start at the top with 2 options, body covered with scales or body covered with hair. Since our specimen is furry, we eliminate scales and follow the path down for hair. That path brings us to our next set of choices, has webbed feet or does not have webbed feet. We match our specimen to the webbed feet description, follow the line to the end, and land directly on our correct answer. By narrowing your focus to specific, observable features, a dichotomous key systematically eliminates incorrect species until only the true identity remains. You now have a complete blueprint to navigate biological classification. You know how to define life with MRS GREN, track evolutionary history with DNA, and identify specific organisms using binomial names and dichotomous keys. The natural world might seem chaotic from the outside, but you now know the rules to read it as a highly organized, predictable system. Armed with these steps and our team to back you up, you have what it takes to break down any IGCSE exam trick question. You've got the basics down, but are you ready for the exam? Visit www.study-buddy.tech to take a free diagnostic test that will allow students to identify their weak areas and become the basis for the creation of a customized study plan for exams.